Alright, today we're going to learn about gaseous change and its control. Okay, so cellular respiration, we use O2 and then we release CO2. So we are using O2 so that we can respire. Okay, O2 is needed by the mitochondria to release energy. For small organisms, they do not need any special organs. They can just absorb O2 and then release it. Take worms, coelenterates. They do not even have organs. Why? Because they are so small, so thin, meaning that they have a high surface area to volume ratio. Okay, so if you look at this picture, there are three organisms. The first one is the smallest one, 1 meter across, 1 meter deep, and 1 meter wide. Okay, so they have six sides. So if you total number of sides, there are six sides. And then if you look at the mass, 1 times 1 times 1, so it equals 1 meter cubed. So 6/1 means that you have 6. So they have 6 times surface area compared to the volume. Now move to the second one, 2 meters across, 2 meters deep, and 2 meters wide. So what does it mean? It means that this animal is much more bigger, and also the surface is not really big enough for the whole volume. Volume is 8, and the actual the actual surface will be 24. So 24/8, you get 3 to 1. So it's little half compared to the smaller one. And then move to the bigger one, 333. So you get 27 meters cubed. What about the size? Size would be, if you look at the surface, is 54. So 54/37 is 2 to 1. Mean that the bigger the animal, and the more tissues that you have in your body, and not all of the tissues are on the surface. So you actually need something to get the O2 into the body of the animal. So for mammalian respiration, you have a tubular system and a pair of spongy lungs. So the spongy lungs would be what we call the alveolus. So alveolus in plural, we call it alveoli. We give a huge surface area for gas exchange, and within these small structures, it is very well adapted to efficient gas exchange. So it is very thin organ. It only has one layer of cells. If you compare it to your skin has a lot of layers, multiple layers, but here only one layer. And why is it so thin? It's so that the diffusion will be easy. The thinner the surface, the easier the diffusion. Around that, you have blood capillaries. So capillaries are small, let's call it small for vessels. Okay, small vessels around that alveolus. So all of these are for sending and receiving gas. So they would send CO2 out, and they would receive O2. Okay, so if you try to blow air into water, you'll have bubbles. So air and water do not mix. O2 and water really cannot mix. So if you look at our blood is made out of 90% water. So how can it carry oxygen? The answer is by using a pigment. So pigment allows O2 to dissolve into the water, which is our blood. So in this case, our pigment is called hemoglobin. Okay, so the vehicle for transporting gas is called hemoglobin. So hemoglobin is made out of four subunits of protein, two alpha, two beta, and each subunit has a heme group and globule protein, and within each heme group is a porphyrin ring with an iron atom. So this group is the one that causes your blood to be red. Okay, so here's the molecule. You have one, two, three, four, two alpha, two betas. Each one of these chains would have their own heme group, and in the middle of the heme group you have one metal ion, which is iron. Okay, so hemoglobin has four heme groups. They can bind reversibly to one O2 molecule without reacting with it. Means that once it binds with oxygen, you can re-release it. If you compare with other chemical reactions with oxygen, calcium plus oxygen becomes calcium oxide. Ferrum becomes ferrum oxide. Okay, ferrum oxide means that you have rust. Iron plus oxygen, you have rust. But in this case, for hemoglobin, it binds to oxygen for a while, and then it will just be releasing it. Okay, so as you can see, the equation will be Hb plus 4O2 is HbO8. Okay, so each red blood cell contains 250 million molecules of Hb, meaning that one Hb would carry 4 molecules of oxygen. So times by 4, you get 10 million. I mean, sorry, um, 10 trillion O2 molecules. To increase the capacity for transporting O2 molecules, red blood cells do not have any nuclei, and also the shape is kind of like a flattened disc. It's not sphere, so that you can actually carry a lot of oxygen, and also it can stack. Okay, so if you look at the red blood cells, they look like discs. So if they are flat like that, they can stack. When they stack, mean that within your capillary, they can actually um, they can be stacked together a lot. They can carry a lot of oxygen that way. Okay, so as you can see here, this is the blood of a diabetic sufferer. Okay, so sugar, if they are not controlled, it will not be converted to glycogen, and due to that, it will just be floating in the blood. And if they are floating in the blood, it can actually get into your red blood cell. Your red blood cell, instead of binding with oxygen, it will actually bind with sugar. So what we'll have is glycated hemoglobin, HbA1c. So it means that your red blood cell would not be really good in carrying oxygen. I mean that people with diabetes, they will be having shortness of breath, uh, tiredness. Okay, so meaning that it will actually cause you to be to lack oxygen all the time. High efficiency in HB as a respiratory pigment in binding O2 is due to its affinity. Affinity means that it's like a favoritism, means that you really like something, you go to that thing first. So in this case, hemoglobin would really easily bind to O2 even at um, low volume. Okay, yeah, so the thing is, you're not actually breathing in 100% oxygen. Okay, Earth is like 21% oxygen, but that's enough for us. Okay, the other 70% will be nitrogen gas, other gases, but somehow we are able to survive in this Earth. Why? Because hemoglobin is so good. It is so it is so high affinity in O2. So imagine if we are living in a another planet with even 10% O2, we still can survive. Okay, so hemoglobin can really easily um, release O2 when there's a drop in O2 itself. HB has high affinity for O2 when PO2 is high in the lung, and low affinity for O2 when you are in the respiring tissues, meaning that when in the lungs, it will really easily soak up all of the O2, and then when it goes to your tissues, it will just really easily release the O2 to give it to your starving cells and tissues. Okay, so now we are learning about diffusion. But in this case, diffusion is involving a gas. So we're using some terms called partial pressure. And so partial pressure means you have this gas with its own pressure versus gas with another gas with another pressure. So within the lungs, when you breathe in the air, you have a lot of partial pressure of O2 in your lungs versus your blood vessel. In this case, in terms of diffusion, high goes to low. So the high oxygen in your lungs goes into your red blood cell, which is low in partial pressure. Okay. Conversely, you have CO2, which is high in the red blood cells. Okay. when it reaches the lungs, there is a very low CO2 in the lungs, so it will go from red blood cell to the lungs because of the difference in uh, rep- because of the difference in partial pressure. Okay, so let's continue. Hb has high affinity in O2. Okay, we have done here. Okay, so the speed of which blood transport O2 to supply the requirement of the body relies on the high affinity of Hb for O2. So you have Hb plus O2 and to carry it all the way to the body. Okay, and then you become oxyhemoglobin, and due and then when it goes to the cell, HbO8 will become Hb plus O2, means that O2 will release. So you have this small pigment that is able to combine with oxygen, okay, without reacting, and then you'll be able to release it. Yeah. So the speed to release it or to hold on, it actually depends on where the red blood cell is. So we have the oxygen dissociation curve. Okay, so as you can see in this curve, it is kind of like a hill. Okay, so on the y-axis you have the SaO2, and the x-axis you have the PaO2. SaO2 means how much oxygen that is in your blood. PO2, which is the pressure of oxygen, it is the pressure of oxygen in the lungs. Okay, and so you have SO2, PO2. So with this graph, it actually shows you that even at small drops of oxygen that is in the tissue or in the blood, oxygen is released. Okay, even though the cell is not really active, but still a red blood cell would give the oxygen. Okay. Other than that, if you can see, um, the saturation declines steeply as PO2 falls, meaning that as you are far away from the lungs, it will be easier for the blood to be saturated with oxygen. Okay, so what are the other factors that is in your body that causes oxygen to be released faster? So when you are doing active, active activities, movements or whatever, you will be releasing a lot of CO2. The more CO2 released, mean that your tissue will be needing more energy, and due to that, your oxygen delivery or transport will be even faster. Yeah, so mean that when PCO2 in blood increases, affinity of HbO2 decreases. So the graph will actually go all the way to the right, meaning that you'll be releasing even more. Okay. Other than that, the effect of pH. So PCO2 or partial pressure of CO2, it is it also causes your blood to be a little bit acidic. So the more acidic your blood is, the easier the CO2 will be released. Okay, and so both acidic and alkaline in your blood. So whenever there is a lot of CO2 in your blood, it will actually causes your blood to be a little bit acidic. So this should not be prolonged. So what happens that during the transport of oxygen, your red blood cell and your hemoglobin will actually um, take CO2, and at the same time it will be able to neutralize the acidity that is within the blood. Okay, so you have H plus from the from the acid. It is bound. It is actually bound to Hb. Okay, so Hb or hemoglobin, um, they came from the lungs carrying oxygen, but uh, when they come to the tissue that need oxygen, usually that tissue will have CO2. Okay, so they would exchange. They will exchange O2 to be released, and then they will grab the H plus to make it neutral again. Okay, so remember in chemistry, if if we have a lot of H plus or protons in the water, in aqueous solution, it actually causes the solution to be acidic. So in this case, your red blood cell is able to actually couple that H plus with HCO3, okay, HCO3 in blood. So it actually causes you to have CO2 and water. Okay, so when you delete the H plus in any solution, mean that the water will become from acidic to neutral. Okay, so when tissue metabolic rate increases, you have more CO2. CO2 form bicarbonate ions and protons. You see CO2 plus H2O become H2CO3 and then H plus and HCO3. So this H plus needs to be neutralized. Okay, so what will happen is that Hb would release O2 and grab that H plus to become Hb. Okay. And in the lungs, the reverse will happen. Hb, okay, the H will be combined with HO3 to form water and CO2, and Hb or hemoglobin will grab oxygen instead. So both of these happen at the same time. There will be little change in blood pH. Okay, so the Bohr effect, it is the results of O2 shifting to the right when CO2 or H plus acidity increase. More acidity, the more oxygen will be released. Okay, because your body needs to survive. If your body is too acidic, then multiple organs will fail. Okay, so as you can see in these animals, some animals would have myoglobin instead of hemoglobin. So myoglobin is this special type of pigment that is found on active animals. So when I compare with hemoglobin, myoglobin only has one heme group. Hemoglobin has four heme groups. Okay, it means that the hemoglobin is like a four-seater car. You can have four passengers, but myoglobin is just a motorbike. Okay, just the one passenger, meaning that it's much more faster, it's much more rapid. Okay, if you look at this graph, the more active the animal is, the more myoglobin that it has. True seals would have a lot of myoglobin because it needs to swim. It also needs to heat up itself. Because it lives in very cold water. It is a mammal actually. And then what else? We have the pig. Okay, the pig, they are not really active. They just they're not running everywhere. So a lot of they have very low myoglobin. So beavers would be in the middle of it. Okay, and so as you can see here, um, the animals which have a lot of myoglobin, the meat will be red and rich. Okay. Um, Other than that, when you try to cook them, usually the oxidation of myoglobin will make it turn brown from red to pink to brown. Okay. So what causes the color of the meat? It will be hemes. Okay. So if we want to compare, what about humans? Humans also have myoglobin. So you find it in type 1 muscle, type A, and type B. Okay. So type 1 muscle is the muscles of the marathon runners, long-term runners, mean that they are able to sustain the movement for long distances and also for a long time. And if you want to look at type B, 2B, 2B are sprinters. Okay, a lot of power, but for a short time. Okay, and then 2A will be in between. So all of these people, they have myoglobin, especially in the legs. Okay, so as you can see here, the myoglobin's graph is much more closer to the y-axis compared to hemoglobin, meaning that will be easily releasing the O2 to tissues, even at low partial pressures. Meaning that if you have myoglobin, it'll be really hard for you to feel out of breath. You'll always be oxidated. Okay, so that's why if you look at athletes, athletes can run for a long time, and they can perform for a long time, and even after that they can still they can still do work. If you want to compare with us, yeah, just regular people, if we just ask us to run a marathon, after the marathon we will just be so tired that we sleep. But for them, they still have enough oxygen to function. Okay, so as you can see here, on the left you have the hemoglobin molecule. It's kind of like a big molecule with four hem groups. Myoglobin is the one on the right with just one hem group. Okay, so if you look at the function, they function the same. They carry oxygen. It's just that oxyhemoglobin, they release O2 at very low pO, meaning that O2 can store in resting muscles and help delay the onset of anaerobic respiration during workout activity. And so, if you want to compare with hemoglobin, hemoglobin is much more generous. They will release oxygen easily, but oxymyoglobin in us, we don't actually use it a lot. Okay, so they just stay in your muscles. It's just used when you are in emergency situations. Okay. CO2 is produced from aerobic respiration cells, either diffused directly or into tissue fluid, and it goes to the lungs in three ways: in plasma, in hemoglobin, and in conversion into hydrogen carbonate ions. Okay, so conversion to hydrogen carbon ions (RBC). So we have 85% of the CO2 released during respiration will be turned into HCO3 minus and H plus. H plus will be absorbed by the hemoglobin, and HCO3 will just be floating in the blood, in the fluid of the blood. Okay, more acidity will be controlled by HB, and then you know the exchange will happen. O2 will be released by Hb. Hb will take in or H plus ion, protons. Okay, so this is what you call the Bohr effect. So twin plasma will diffuse and combine with water to form H2CO3, and then H2CO3 will release H plus and HCO3 minus, and this H plus will be exchanged with O2 by hemoglobin, and hemoglobin will release oxygen. So at this point, the acidity of the blood will be neutralized. Okay, so if you can see, venous blood, you have CO2 being released, and it gets into the red blood cell, and then this red blood cell is transported to lungs, which causes the CO2 to be released into lungs, and then um, converted into CO2 gas and water. Okay. Some of them also becomes hydrogen carbonates. Okay, these hydrogen carbonates accumulate in RBC, which is plasma to form hydrogen carbonate, sodium hydrogen carbonate with sodium. So free HCO3 will be carried to lungs and converted to CO2, and in the lungs it will just be breathed out. Negatively charged CO2 minus will be diffused out to plasma, and it will become positively charged due to accumulation of Na plus. And because of that, the red blood cell needs to neutralize the cell, so it needs to absorb Cl minus ion from plasma into cells. So this is actually called the chloride shift. And so um, to actually to actually cause neutrality, um, your blood needs to actually have a lot of salt ions at all times. So your blood is healthy is due to that. Okay. Okay. So 10% of the CO2 will actually combine with hemoglobin to form carbamino hemoglobin. So just like O2, hemoglobin will latch onto them and then carry them all the way to the lungs, and then in the lungs it will just be released. So other than that, if you look at your soft drinks, in soft drinks, there's a lot of CO2 dissolved in that solution. So same thing goes in for the 5% of your CO2. 5% of the CO2 actually dissolves in your blood as what you call it H2CO3 or carbonic acid. So carbonic acid, um, they are just in a solution. Okay, Sometimes they can just diffuse into H plus and H3 minus. Okay. So this is actually causing the blood to become acidic, but your plasma protein will buffer it. Proteins, they're able to absorb H plus. Okay, they will actually cause it to become neutral again. Okay, H3 minus transported by the blood to the lungs and then converted to CO2 and water. Okay, so in the lungs, PO2 is high, PCO2 is low. O2 will diffuse into alveolar blood capillaries from alveoli, and CO2 will be diffusing out from the alveolar blood capillaries into alveoli and expelled in the air. So you can see the exchange happening. O2 from lungs into blood, CO2 from blood into the lungs. So now we move to plants. So plants, they need CO2 to build up their tissues to build up glucose by photosynthesis. But at the same time, they also have to do respiration. For aquatic plants, where do they get CO2? It is from the hydrogen carbonate ions, HO3 minus and carbonic acid in the water. Okay. What about terrestrial plants? CO2 is in the air. They can just get it from the atmosphere, just like that. So they only have one organ that is responsible for this, called the stomata. Stomata is just a pore or a hole under the leaves where the air can come in and out. Okay, so stomata is found much more in the bottom of the leaf compared to the top of the leaf because they want to reduce the loss of water. Um, Stomata is important because they are the only place where gases can come in and out. So it is for exchange of gases, and O2 produced will be diffused out of the leaves, and the CO2 in oxygen in the atmosphere will get into the leaves for photosynthesis. Okay, so the holes within the stomata is controlled by two cells, which is called the gut cells. So these cells, they look like beans because they're kind of like uneven in terms of their thickness. The outer part will be less thick. The inner part will be much more thicker. Okay, so usually for plant cells, when they absorb water, they will become turgid, and then when they lose water, they become flaccid. So what controls the opening and closing of stomata will be the turgidity and the flaccidity. If there's a lot of water, they'll close. If they lose water, they'll open. Okay. Okay. So opening and closing the stomata is in response of increase and decrease of water potential accept solution and in gut cells. So usually when you have photosynthesis happening, photosynthesis will actually cause a lot of glucose to be created. Glucose or sugar, they will cause any solution to become much more hypertonic. When they're hypertonic, they will absorb water from the surroundings, causing it to become turgid. If there is no sun or no photosynthesis, so a lot of this sugar will be turned into starch. So starch is really not osmo. It doesn't have any osmotic power, so it will actually cause the solution to become hypotonic instead. Okay, so actually water in gut cells increases water potential, causes it to become turgid. Okay, and then loss of water will make it become flaccid. So to explain this, we have the starch sugar hypothesis. Okay, so as you can see here, these bean-shaped cells, okay, they do photosynthesis during the day, and then a lot of sugar will be created, a lot of glucose will be created, causing it to become um, much more hypotonic. When they're hypertonic, they will actually a lot of suck in a lot of water, and then they'll be turgid. When they're turgid, they will open. Okay, okay. So during the night, what happens is that a lot of the sugar will turn into starch. Starch is almost osmotically inactive, so it will become hypotonic. Hypotonicity means that you lose the water. Water will be lost, and the cell will become flaccid. So when a cell becomes flaccid, usually they will just lose their toughness, lose their shape, Same thing goes to the stomata. Stomata cell, gut cell will actually become flaccid, and then the hole will close. Okay, so this is the mechanism of stomata opening closing. You have open stomata. To close stomata, you need starch up, water pressure up, gut cell flaccid. Okay, from close to open, you need glucose up, water potential down, and gut cells turgid. Okay, so um, what about the factors affecting the opening and closing? So we must remember that all of the cells that are on the leaves, they are able to do photosynthesis. So if the light that hits the stomata cell is a light that is conducive, then it will do more photosynthesis, mean that it will cause more opening. Okay, so if you look at the leaves they are green because they reflect green light. Okay, mean that other colors rather than green are absorbed. If you shine them with green light, they will not open. They will just be reflected. But for blue and red light, it is the best light for photosynthesis, mean that they will open the widest. Okay, so if the gut cells were exposed to light which are appropriate, they will open because they will do photosynthesis. They will have a lot of water potential. They will be hypertonic. They will suck in water. They become turgid, and then they will open. Okay. When exposed to inappropriate light, they will not do any photosynthesis. What will they do? They will just turn that glucose into sugar, into starch, and then they will just lose all the water, become hypotonic, and then they will just become flaccid. Okay. So um, what about pH? If you wanna compare it with humans, it is similar to humans because um, anytime you have CO2 in something, it will actually cause that thing to become much more acidic. Okay, so when CO2 decreases due to photosynthesis, the leaf pH rises. It means that the leaf becomes much more alkaline. The opening and closing stomata is sensitive to changes in leaf and pH, in pH of leaf. High pH should simulate stomata to open, and low pH will cause it to close. So you can see the similarities here. Alkaline, open. Um, Acid, close. Okay, so increase in leaf pH is followed by decrease in starch content and increase in glucose content in leaves. Okay, so what does it mean? It means that when a leaf is having as low CO2, okay, or much more alkaline, mean that it needs more carbon, it needs more food, so it has to do photosynthesis. Okay, so such glucose equilibrium is shifted right due to pH sensitive enzymes. So phosphorylase, it converts starch to glucose. So phosphorylase actually needs a neutral pH, pH 7. So if it becomes acidic, then phosphorylase will not work. It will not be able to turn starch into glucose to open that stomata opening. So when leaf pH drops, it becomes more acidic, meaning that it's not really suitable for that enzyme to work. So the starch glucose equilibrium will be shifted to the left. It means that the leaf already has enough food, enough oxygen, enough CO2, so it will actually cause a lot of the glucose to be turned to starch, and then because of that, the the gut cells will lose water, it will lose turgidity, and then it will close. So that's all for today. The end.